So this lecture is an introduction to enzymes and enzymatic catalysis. So before we begin talking anything in any significant depth about enzymes, we want to define them, what they are and what they do in the cell. So enzymes, first and foremost, are proteins. So primary, secondary, tertiary, and quaternary structure are basically enzymes. These are proteins that catalyze reactions in the cell. So anywhere from 50,000 to 100,000 enzymes, which are also proteins, are found in a typical cell. So you may have heard in biology class, enzymes are catalysts, and so they're going to do everything that a catalyst does. You'll find that an enzyme will take a reaction and do it at a much faster rate than we could ever do it in an organic chemistry lab or in any chemistry lab. So enzymes are remarkably efficient. If you remember, catalysts lower the activation energy of a reaction, and that's the basis for which they enhance the generation of products from reactants. We don't have reactants going to products like in general chemistry. What we have is the enzyme taking a substrate. S stands for substrate. We have this intermediate known as the enzyme substrate complex, and that gets released into the enzyme and free product. It's very important that you realize that the enzyme must be free to catalyze a second, third, fourth, fifth, however many rounds. So you need to regenerate the original enzyme in its original three-dimensional shape. So the abbreviations for here are the substrate. The substrate is something that's going to get converted into product. Now, enzymes being these big proteins, the substrate's going to bind to a very specific part of the protein in the enzyme, and we call that the active site. That's where chemistry happens. That's where your organic chemistry mechanisms, E1, E2, SN1, SN2, acid base, that's where all of the chemistry happens at the active site. So some amino acid residues are going to be important in the active site. So catalysts are what enzymes are, but enzymes are much better than normal catalysts that you can find in general chemistry lab. The reaction rate is faster, anywhere from 10 to the 6th to 10 to the 7th rate enhancement with an enzyme than without an enzyme. The enzyme has to create a substrate to product within the confines of the cell. So remember, physiologic pH. We can't play around too much with temperature or pressure. So in that context, enzymes are much better than the organic chemists, organic chemistry synthesists, or the people who synthesize drugs and compounds, because they can play around with pressure and temperature and make a reaction go under harsh conditions. Enzymes don't have that luxury. They have to do it in the conditions inside the cell. Enzymes in this third bullet point are remarkably specific. You don't want to hold too tightly to that bullet point, because we do have ways, which are drugs, that can trick an enzyme into taking something that is other than its cognate substrate. So even though enzymes are very remarkably specific for the substrate, in some cases discriminating between a DL stereoisomer. In some cases, this is all drug therapy actually, and drugs. We find ways to actually exploit a substrate to make it look like it's real substrate and trick an enzyme, and that way kill an enzyme. So this whole thing deals with the properties of inhibitors, which we'll talk about also later in this module. Enzymes can be regulated. So unlike organic chemistry lab or any synthesis lab, enzymes can be modulated. An enzyme can be at full force, ready, gung-ho, or an enzyme can slow down, or an enzyme could be somewhere in between. It's really accustomed to the physiologic state of the cell. So you can imagine the enzymes in glycolysis when an individual is undergoing rapid exercise and full exertion. Those enzymes are really on. In the case of someone who's sitting down and resting, maybe the enzymes of glycolysis or any enzyme are at a very basal level. Okay, so very running, but running in the background. So that's very powerful in the sense that enzymes can be controlled. It can also be regulated. You can turn off or turn on an enzyme called that allostery, and that's something enzymes can do that we cannot do in the organic chemistry synthesis lab. So enzymes are very, very specific. They react within the confines of the cell. And this last bullet point is a very important bullet point, even though it's kind of minimized and relegated to the bottom. Enzymes sometimes need help. So enzymes need help in the form of cofactors. Enzymes need help in the form of prosthetic groups. So you're going to see a lot of enzymes that are going to require what are called cofactors. How good is an enzyme? Shown in this table from your textbook are 6 enzymes: carbonic anhydrase, corsmate mutase. This enzyme is important in the metabolism of phenylalanine and tyrosine. Triose phosphate isomerase. This enzyme is important in glycolysis, one of the steps in glycolysis. Enzymes that break apart proteins, enzymes that break apart nucleic acid. Want to just let you know, a lot of enzymes by default end in the 3 letters ASE, ACE. So you can have ligases, proteases, dehydrogenases. So this large umbrella term known as enzymes, for the most part, a lot of them end in the term ASC for ACE. Now look at the non-enzymatic reaction rate. Carbonic anhydrase will catalyze the reaction of CO2 and water, giving you carbonic acid. So this happens in our atmosphere or by itself very, very slow, okay? But put it in carbonic anhydrase, whose 3-dimensional structure is shown here, and you get a millionfold increase in the rate. Actually, more than a millionfold. Actually, this is about 8 millionfold enhancement of carbon dioxide and water forming carbonic acid. In the presence of this enzyme, carbonic anhydrase, you get 8 million times the reaction occurring. So that's the power of these enzymes. So you can actually see here the rate enhancements are extraordinarily large in the presence of an enzyme, whereas without the presence of the enzyme, just the reactants by themselves, you're getting nowhere. In some cases, for these nucleases and nucleosidases, 10 to the minus 11, 10 to the minus 13, that's virtually nothing. But if we're going to rate enzymes on their efficiency of converting substrate to product, we have a lot of parameters. Two of the more important parameters are Km and Vmax. Km stands for the Michaelis constant, and Vmax stands for the maximum velocity. Showing you some proteins. So if you love structures, enzymes, which are the powerhouses, the workhorses of the cell, converting substrates to products, are basically folded proteins, just all amino acids. And some of these are, here you have triose phosphate isomerase, sort of related by some symmetry. Some of these are related as tetrameric. Some of these are just monomers, such as carbonic anhydrase. So all of these structures are what I got and rendered in the protein data bank. Might as well just hit it now. How good is an enzyme? Right, so 2 parameters are Km and Vmax. So shown here is the Michaelis plot, or Michaelis-Menten plot. Shown is the velocity of an enzyme, how fast it converts a substrate to a product, and also on the x-axis would be the substrate concentration. We're going to define something called Km, Michaelis constant, and that is the substrate concentration where V is equal to Vmax over 2. So let's get that into our heads now. Let's concretize and solidify this very important parameter, Km, standing for the Michaelis constant. It is actually a substrate concentration. It is a substrate concentration where V is equal to Vmax over 2. So if you think about this, if you have a low Km for an enzyme, talking about enzyme performance, if an enzyme has a very low value of Km, it means it takes a low substrate concentration to get to Vmax over 2. The substrate has a very high affinity for the enzyme's active site. Remember, the active site being where chemistry happens. Now, on the flip side, if you have a Km parameter that's very high, in a numeric value, it means it takes a high amount of substrate to get to Vmax over 2. That implies that the substrate has a very low affinity for the enzyme's active site. So Km and the affinity of the substrate to the active site are intimately related. Typical profile for a Michaelis plot where velocity is on the y, substrate concentration on the x, is your hyperbolic plot. We saw this with myoglobin binding to oxygen. We also see that with substrate being converted to a product. And of course, the asymptote here is the Vmax, something we want to achieve, but we really only achieve that if we get to very, very high substrate concentrations, really saturating that enzyme's active site with substrate. So what are these enzymes? They're everywhere in our cell. Here's an enzyme, corsmate mutase. It's going to convert this intermediate into that. I do not know the names of this compound and that compound, but I do know it's involved in the metabolism of the aromatic amino acid tyrosine and phenylalanine. Look, it has a three-dimensional structure. Somewhere in the vicinity of the enzyme is an active site where this conversion happens. Notice the double-sided arrows. So a lot of enzymatic reactions can be at an equilibrium. It can go forward or it can go backward, dependent upon the needs of the cell, dependent upon the flux of the metabolic pathway. Here's another enzyme, triose phosphate isomerase. So if we ever have an opportunity to study glycolysis, you will be very familiar with this enzyme, and it's going to convert, so triose phosphate isomerase is going to convert dihydroxyacetone phosphate, shown here on the right, to glyceraldehyde 3-phosphate, shown on the left. Okay, it's a free interchange. Triose phosphate isomerase catalyzes that reaction. Interesting bit of trivia on triose phosphate isomerase. Some enzymologists dub this enzyme as a catalytically perfect enzyme. So what do I mean by catalytically perfect enzyme? It does the reaction very, very fast. In terms of all you need is the substrate to get into the enzyme's active site, and bam, you get the product. Okay, whether it's this product here, again, dihydroxyacetone phosphate, or this product here, glyceraldehyde 3-phosphate, do not need to know those names. It's just very, very fast, and we call that a catalytically efficient enzyme. Again, performance metrics, how good is an enzyme? Oftentimes confusing, but we can classify the different reactions an enzyme catalyzes based upon what they do, the chemistry that they do. So oxidoreductases are going to catalyze redox reactions. Examples are the dehydrogenases. It's a classic example. Dehydrogenases, usually what they'll do is they'll take a cofactor, NAD+, and reduce it to NADH, or that cofactor can be FAD+, and they'll reduce it to FADH. So some oxidation, some reduction reactions. Transferases are just going to transfer groups. So here, the hydroxymethyl transferases are going to transfer different types of groups. Usually they require cofactors. In this specific example, we require the cofactor folate. Okay, this is why folate is needed in our body. Also realize these transferases are important in amino acid conversions. Okay, in this reaction, you can convert serine to glycine with these transferase reactions. Hydrolases will do hydrolysis reactions, so urea can get split by urease into CO2 and ammonia, and the urease, which is a hydrolase, catalyzes that reaction. Lyases will cut apart bonds. Isomerases will isomerize. Ligases will ligate, join two things together. But enzymes require help, some cases. Sometimes enzymes require metals. So remember the copper zinc superoxide dismutases. Metals, some enzymes require that. Some enzymes require these reducing equivalents, NADH, NADPH, nicotinamide, adenine dinucleotide is what NADH stands for. Flavin, adenine dinucleotide, the flavins, that's what FADH stands for. Heme, some enzymes do require heme. Some enzymes require even more complicated cofactors, like lipoic acid. Biotin is important in enzymes that require carboxylation. Here are the very famous NADP or NADH cofactors. You'll see them a ton in dehydrogenation reactions. Here's tetrahydrofolate. So a lot of enzymes require folate or folic acid. In fact, deficiency of folic acid or folate is very, very bad for the health, especially for a developing embryo. Here are the flavin. Adenine dinucleotides, important in electron transport chain, so they're going to interact with those enzymes. Remember that when the substrate gets converted to product and chemistry happens inside the active site of an enzyme, which is a protein, the enzyme also must have binding or accommodation sites for these cofactors as well. So a lot of diseases can manifest themselves, for example, if biotin is not lodged correctly into the active site or does not have access to the active site. So that's a very important point for us to realize. It's not just the protein and the active site amino acids that are important, but making sure that these cofactors, if the enzyme requires a cofactor, it's important that these cofactors are also put in the proper orientation and place within the active site of the enzyme. Even more cofactors here, vitamin B12. There's a disease called pernicious anemia. People with pernicious anemia do not have adequate vitamin B12. It does not get into the enzyme that is involved in that pathway. Diamine pyrophosphate is a cofactor. You'll see this if we study biochemistry further. This is very important. Thiamine is in the pyruvate dehydrogenase multi-enzyme complex, something that we will study later on in the course. Deficiencies in thiamine cause beriberi. It's a disease prominent in third world developing countries. Also in adults, people who are alcoholic and do not have adequate thiamine, they also suffer from a disease. It's called Wernicke-Korsakoff syndrome. Pyridoxal phosphate, this is a vitamin B6 derivative. Pyridoxal phosphate is a very important cofactor in transaminases, which are important in the liver. They are intricately involved in converting an amino acid to an alpha keto acid. So just giving you an idea of the importance of all these different types of cofactors. Main point, enzymes are proteins, but they need help. Sometimes the help comes in the form of metals. Sometimes the help comes in the form of all these different types of cofactors. A couple of more, iron. So here we have what's called an iron-sulfur cluster. You'll see that in many of the complexes of the electron transport chain, and obviously some of the metals that we alluded to before. For example, zinc. A lot of times zinc gets coordinated with histidines. Iron, we also saw when we studied hemoglobin, gets coordinated by a proximal and distal histidine. So the role of the metal not only facilitates chemistry, but it has to be in a right spot and coordinate with very conserved amino acids. So here's an example of an enzyme that requires a cofactor, alcohol dehydrogenase. Ethanol gets converted to acetaldehyde. In that process, NAD plus gets reduced to NADH. So a very important enzyme. Methanol is a competitive inhibitor of this enzyme, and a very interesting story with regards to methanol blindness. So here's the structure of NAD plus. And then here's the reduced form. NADH is a very, very important reducing factor in the cell, and it's very good that the cell produces these reducing equivalents. The NADH can also go in and participate in other dehydrogenation reactions, but dehydrogenase is an example of a reaction that requires a cofactor. And number two, as all dehydrogenases do, they are redox reactions, so oxidation, reduction. Enzymes are very specific and discriminatory, but don't hold so fast and steady to this. Like I said before, a lot of pharmaceutical companies design drugs to sort of go into the enzyme's active site and trick it so that it produces something else, or it just kills the virus or kills the bacteria. So that's where we talk about inhibition. competitive inhibitors, non-competitive inhibitors, uncompetitive inhibitors. But the classic sense of the enzyme, yes, they're very discriminatory, they're very specific, and they can discriminate between D or L stereochemistry. Just to show you here, substrate binds to a very specific site in the enzyme, and that's called the active site. Or two models of how a substrate binds to the active site. You can go to your textbook and actually they'll dedicate several pages to these two modes or two models. I'm just going to briefly touch up on it and give you the nuts and bolts. The nuts and bolts are this. We have the lock and key model where the substrate just literally fits into the enzyme's active site as a key would fit into the lock. Very specific. There is no adjustments. There are no conformational changes. It just fits in nicely. Now, contrast that with the second model of an enzyme binding to the substrate, and that is called the induced fit model. So here the substrate binds to the enzyme's active site, but it's not such a perfect like a key and a lock sort of interaction. There has to be some sort of conformational changes. So the enzyme shifts in conformation, some of the structure shifts, and then we get the substrate gets converted to products. So here the difference between the two models is one is fits very nicely. The second model, there are substantial conformational changes. The best example of this is actually several enzymes, but one of them is carboxypeptidase. Also another enzyme is hexokinase, the very first enzyme in glycolysis. Very classic examples of induced fit. So what do I mean by induced fit? So here's your substrate, here's the active site, and like Pac-Man, the substrate grabs onto it, or it clasps the substrate. So a lot of conformational change. You can imagine a lot of shift in the enzyme's tertiary structure and secondary structure. Some enzymes, as we stated before, are specific, but some enzymes are a little bit more permissive, such as your digestive enzymes. So these are proteases. Chymotrypsin, trypsin, pepsin are your classic proteases. They're going to cleave peptide bonds. Chymotrypsin in our gut actually can cleave a peptide bond, but sometimes it can also cleave an ester bond. Enzymes are very specific, but don't hold so tightly as that being so dogmatic. A lot of times they can be tricked, or a lot of times they can cleave different types or very similar types of bonds. Enzymes are proteins. Enzymes catalyze reactions in the cell, converting substrate to products. But going back to general chemistry, these are catalysts. These are protein catalysts. And what does a catalyst do? It lowers the activation energy. So enzymes do this much better than an organic chemist can synthesize a compound in the lab. It can do it with a better yield, less byproducts, less waste, more efficient, and it does it within the macromolecular crowding of the cell where the pH is usually physiologic pH. So this is the remarkable aspect about enzymes. And next thing we want to really talk about in this brief introduction that I've just completed, next thing we want to talk about is really enzyme performance and enzyme kinetics. How can we tell an enzyme is good? Is the enzyme a really fast, souped-up muscle car, or is an enzyme very slow lemon of a car? To do that, we have to define some parameters, such as Km and Vmax and Kcat. The second discussion I want to talk about with regards to enzymes is also drugs. So a lot of drugs we take target very specific enzymes, or they'll target very specific viruses, such as the protease inhibitors in HIV. So we want to talk about how certain drugs can inhibit enzymes in a way that sort of promotes health. Different types of inhibitors, competitive inhibitors, non-competitive inhibitors, uncompetitive inhibitors, and how do they affect the thermodynamic parameters Km, Vmax, and Kcat.